and welcome to the Director's Desk, the podcast where we spotlight the groundbreaking work of the National Institutes of Health and the remarkable people driving innovation in biomedical research and public health. I'm Dr. J. Bhattacharya, the director of the NIH, and joining us today is Dr. Nicole Kleinstreuer, Acting Deputy NIH Director for Program Coordination, Planning, and Strategic Initiatives, which is a real mouthful. It's great to have you on the podcast with us, Nicole, and welcome to the Director's Desk. Thanks for being here. I've been looking forward to this podcast for a while because you lead an incredibly important office. It has a name that I will never be able to remember, I have to say. And inside the lingo of the NIH, it's DP KIPSI. DP
right,
KIPSI.
Yeah.
Did I get that right?
Poughkeepsie.
I've only been here a few months.
Just think about like a neighborhood in upstate New York. Poughkeepsie.
Okay, just a quick test. Can you actually tell us what the actual name of the office that you lead is?
The Division of Program Coordination, Planning, and Strategic Initiatives. DPCPSI.
Dude, I'm sorry, but that's just too much for me.
Well, I have a real affinity for complicated acronyms. That has basically defined my path in government.
I've learned that about you, Nicole. But you also have done a lot of incredible work on promoting alternatives to animal use inside the NIH. That's the main focus today of today's podcast, and I wanted to tell people about that work. Have you tell people about that work? Why don't we start from the beginning? So not quite that far. Why don't we start from your career? So how did you get? What was your training? How did you come to the NIH? Tell us about that.
Sure. So my family has played a huge role in getting me to where I am right now. So my mom is an artist and my dad is a scientist. So he's a biomedical engineer. And from a very young age, I displayed a real aptitude for science and mathematics, and particularly in the area of how can we use those tools to better understand human health and disease, and how humans interact with the world around us, and how can we optimize our experience in this world, and how can we optimize human health for the greater public good. And so that's really what drove me throughout my undergraduate career, my graduate work. So I have degrees in applied mathematical modeling and biomedical engineering. My PhD is in bioengineering. I ended up doing a postdoctoral fellowship in computational toxicology, which was a brand new field at the time. And
so wait, what is that about?
So that's about taking computer models and using them to understand how things like environmental chemicals, toxins, food ingredients, personal care product ingredients, how those types of chemicals can interact with your body and potentially cause toxic effects, and doing it without using animals. Because at the end of the day, we're not 150-pound rats. Our biology is very, very different. And
200-pound rat in my case.
Okay, 140-pound rat in my case. while we're oversharing. No, but computational toxicology is a field that was actually predicated by toxicogenomics, so using things like high-throughput screening and looking at the effects of hundreds, if not thousands, of chemicals on different molecular and cellular targets, and then using advanced computational techniques to build predictive models for how those chemicals are going to affect key biological pathways.
So like just to emphasize, so what you're saying is that rather than exposing a mouse or a rat to some toxic chemical and seeing how, that's often what scientists sometimes do to understand toxic effects of things. Instead, you can build computer models that better predict what would happen in humans if you expose them to the same chemical.
Absolutely. And not only do they better predict what's going to happen in humans because they're built on human biology and human gene and cell targets, but they can do it at scale in a way that is so much more rapid, so much more efficient, so much more so much less resource intensive than feeding a chemical to a group of rats and waiting to see what dose, causes adverse effects or kills them, and then trying to extrapolate that to humans. It sounds so prehistoric because it is. It's how toxicology was done for decades before we had the tools that we have available to us now. And now that we have tools using human cell-based systems, using computer models to integrate that data, and using things like systems biology approaches to really represent human pathways, now that those tools are available to us, we have an ethical and scientific responsibility to be using that best-in-class science to predict effects of environmental toxicants on human health.
So you did a PhD on this topic.
Actually, my PhD was on mathematical modeling of human biological systems. I built what could essentially be like a control system for an artificial kidney. I actually didn't know that I was going to end up in toxicology. I thought more I was going to, be a professor like my dad and build math models that would help us understand human biology and help us improve it and help us design, new therapeutics or medical devices. I sort of stumbled into toxicology. Now I know enough about chemistry and toxicology to sort of pretend to be a toxicologist. But most of all, I really applied that sort of biomedical engineering and mathematical modeling mindset to transforming the field of toxicology and ensuring that we don't need to use animals for things like chemical risk assessments.
So, and you've been doing this then since your graduate work.
Okay.
And you went
for 20 years now.
20 years. So you came. So how long have you been at the NIH?
I've been at the NIH for about 10 years. Yeah, I was at the EPA for a number of years, in the U.S. Environmental Protection Agency, in their National Center for Computational Toxicology as part of the federal Tox 21 research program, and that was amazing to sort of be at the forefront of that and build some of the computer models that the EPA now uses to do risk assessments without relying so heavily on animals for things like systemic toxicity or endocrine disruption. And then I worked for a couple years at a small contract research organization, but still supporting the federal government. You never really leave the federal government once you start there.
I don't know if that's always true. We'll see.
At least
see.
for me, I've been such a committed civil servant, basically my whole career. And then I came over and I started in kind of the leadership chain of these organizations called NICEDM and ICFAM. I told you I had an affinity for complicated acronyms.
Nicole. We talked about this before. You're have to tell me what is NICEDM?
Okay, so it's like nice ATM, like a friendly cash machine. What it actually stands for is the National Interagency Center for the Evaluation of Alternative Toxicological Methods.
Okay, so break that down.
Test methods now.
So what is that?
It is the U.S. federal resource for alternatives to animal testing. And it is kind of the workhorse that supports this congressionally mandated committee called ICFAM. So ICFAM is, I know, here we go again, ICCVAM, the Interagency Coordinating Committee for the Validation of Alternative Methods.
Okay, so just break this down. So these are federal efforts to promote the use of new technologies so that we don't have to test on animals.
Exactly. And it's not even just to promote the use of, but to actually support the development and the validation of these new methods so that they can be legally defensible, so that they can actually be used in federal agency decision frameworks. And so ICFAM consists of 18 different federal agencies. So being the director of ICFAM uniquely prepared me to lead a division like Deep Poughkeepsie that's comprised of 15 different offices and cross NIH research programs, because I already had a ton of experience.
Why you're so used to that. Okay, so but I want to let me we'll get back to your NIH journey, but I just want to delve a little bit into the justification for alternatives to animals, and I want to focus. Like you said, scientific justification. I've heard the dictum. I don't do lab work for a living. I do epidemiology, health policy, health economics work, which generally involves a lot of computer modeling, a lot of statistics. But my friends who do biomedical laboratory work for a living tell me things like mice lie.
Right.
Mice lie. What they mean by that, if I'm understanding them correctly, is that you can, for instance, cause a mouse to have Alzheimer's. Like we know how to cause a mouse to have Alzheimer's. and we can give the mouse some treatment that cures its Alzheimer's. And yet, when we use that same treatment scaled up to 200 pounds or something, you use it in a human being, and it doesn't work. It doesn't actually prevent or treat the senile dementia that a patient might have. That's pretty common, right?
Yes, it's extremely common. So similar to you, I've never done any animal testing myself. I've never directly overseen any labs that have done animal testing, but I have studied so much of that data and analyzed so much of that data that I know how often animal models mislead us. And so it's a scientific problem, it's an ethical problem, and it's a resource
Because
problem.
we want to
an
make sure
efficiency problem.
when an animal is sacrificed, we want to make sure that sacrifice was not in vain.
Absolutely.
That improved human knowledge in a way that really improved human health.
Well, and we really want to get to the point where we don't need animal sacrifices at all. And some of the examples that you started to touch upon are so relevant in this space. So Alzheimer's is a great one. So mouse models are genetically engineered to overexpress amyloid beta plaque. And so many of the treatments, I mean, billions of dollars have been poured into treatments in mice that look so promising and then fail in humans in clinical trials. Same with things like anti-inflammatory drugs. So, there's examples like CD28 superagonists that showed very promising like anti-inflammatory effects in non-human primates and then caused massive cytokine storms in people with just terrible effects. Cardiovascular treatments are another one. Diabetes, cancer, mouse models, tumors in mice respond very well to things like immune checkpoint inhibitors, and that just doesn't work in human tumors. They're much more heterogeneous and much more complex. And so these animal models, not only are they unethical and time-consuming and resource-intensive, they're also misleading us badly. And so they're actually holding back progress in discovery of human therapeutics and human medical interventions and improving human health. And so that's why I'm so excited, that this initiative that you put out at the end of April, so the Director's Initiative around prioritizing human biology-based models for biomedical research.
Just full credit. That's something that you've been working on for a long time, ever since you came to the NIH. That was one of the main things you wanted to.
It's true. No, my entire career has been dedicated to replacing animal models with much better science, more human relevant and more ethical approaches that better represent human biology and really advance the field.
Can I ask you about the challenges of that inside the NIH? I mean, you were here for 10 years. Why, when you told me about this, I got very excited about it. You showed me the data. I was like very excited as a way to advance human health by reducing the use of animals, asking researchers to rely more on validated methods that do predict what the impact of agents would have on humans. I wondered to myself, why was it so difficult? Like, what challenges did you face inside the NIH to move this forward? I mean, obviously you've thrived inside the NIH environment, so it's not all bad news. But
so there are challenges specific to the NIH, and then there are also challenges that are just endemic across the entire federal government and the entire scientific community that operate on different scales. So animal testing for some people is like a religion. It's something that they've been indoctrinated in. They were trained in a lab where their mentor had a mouse model and that person's mentor had a mouse model. And so they have been sort of fooled into believing that that's the only model that can be used to answer complicated questions about human health and disease. And I understand 40, 50 years ago, when that was the best we had, why people might have chosen to go down that path. It's not a choice that I would have ever made personally, but I understand that. But now we have an absolute responsibility to the public and to the animals, those who can't speak up to defend themselves, to be using the absolute best science available to us to replace animal testing with human relevant methods. The other challenges are not just sort of this scientific dogma and a comfort level with doing things the way we've already done them. The other challenges are the complex landscape of legal and regulatory requirements.
Well, so the FDA just took an action recently. Yes. My friend, Commissioner Marty McCary did it. I always get that name wrong. He's a good friend of mine. I always
just call him Marty.
I always call him Marty. But yeah, Marty McCary got just recently decided, took an action to say that the FDA is no longer requiring or mandating animal testing in certain circumstances when it didn't make sense to do it because it didn't predict what impact those agents would have on human health.
The FDA is taking tremendous leadership in the regulatory space. It has been such a joy to be really intimately engaged with those initiatives. So I was really fortunate to help the FDA in consulting on the development of their strategic roadmap that they managed to get it out a couple weeks before we got our press release out in April. But it's a strategic roadmap on reducing and ultimately eliminating a reliance on animal models for preclinical safety assessment, starting with monoclonal antibodies, but expanding to other medical therapeutics. On Monday, we had this amazing workshop, this joint FDA-NIH workshop on reducing animal testing, and it was transformative. It was really nothing short of revolutionary. It was a meeting unlike any other meeting I've ever seen hosted by the FDA before. They put out so many sort of clear objectives and tangible next steps that they're going to take to change the way they review things like new drug applications and preclinical safety packages. They gave examples from the toxicology space, examples that, my group and other groups in the scientific arena have been working tirelessly on for years and years, demonstrating that there are validated non-animal methods that are superior to sort of the legacy animal tests for endpoints like skin sensitization, skin irritation, pyrogenicity testing. And they walked through those examples as, here's some immediate actions that we can take to start accepting non-animal methods for safety assessments. And it was just so refreshing.
I mean, the nice thing about that is that you have documented data and you're replacing a system where you essentially, if you're a company putting a product out, you're forced to do animal testing.
Right.
And then animal testing doesn't predict what it's going to do in human populations.
And I will tell you, the industry is so ready for this. I mean, the pharmaceutical industry is already using things like tissue chips, microphysiological systems, organoids, computational modeling.
Nicole, I got to actually physically see an organoid the other day for the first time in a little test tube. It's a little tiny, you can see it with the naked eye.
Well, and we are going to be, so under the new initiative and the new office that we intend to stand up, ORIVA, we are going, another acronym.
Okay,
Office
Nicole.
of Research, Innovation, Validation, and Application. We are going to be dramatically expanding
That's the new office you're
infrastructure.
going to be leading inside the NIH, inside the Office of Director.
Inside Deep Poughkeepsie, yeah.
Inside Deep Poughkeepsie. And the goal of the office is to when we do scientific work, to ask researchers to think.
Yes.
Right? Is the animal model that we're using, does it actually predict what's going to happen in human health? Are there alternatives available where you can get better information without having to sacrifice any animals? I mean, we're going to essentially tell researchers all across the country, just think about the work. Make sure that the work that you're doing really does translate to better human health.
I would say that what you just articulated is the goal of the announcement that I made on Monday's workshop with the FDA, where we are saying that all of our new funding opportunities will include this sort of mandate to consider new approach methods and non-animal approaches. And that we will not be putting out any funding opportunities that are, exclusive to animal models anymore. That's the goal of that. The goal of ORIVA goes so much further than that. The ORIVA is to actually create systemic change, to actually enact a cultural shift across the scientific landscape and to put real resources and coordination across all of the excellent efforts that are ongoing in this space. I mean, so many institutes and centers are already doing outstanding work in human relevant new approach methods, NCATS, NIEHS, NCI, NIAID, NHLBI. I mean, pretty much every institute and center across NIH has work in non-animal methods. And so the time is really ripe to have a point of coordination and a nexus to amplify that work and accelerate it and take it to the next level so that it becomes the default rather than the exception. And we are using the best tools available to us to actually provide meaningful, actionable insights into human biology and susceptibility to, all kinds of things.
All right, we're running close to the time here, but I just wanted to make sure that we covered one more thing, one more topic, and this is a sensitive one. It's about dogs and cats, right? So one of the things that I did when we first got here was to essentially say, for research that's done on the NIH campus, we're not going to be doing work with beagles.
No, you shut down all the intramural beagle labs that remain.
Right. So that's that I think was quite popular, and I looked carefully at the work, and I thought, this is this kind of thing where there ought to be alternatives available. It made sense, and of course there were all those terrible stories about beagles from before. So I think it was the right thing to do. What should the NIH do? What policies should we adopt? And I think about dogs and cats going forward for the rest of the portfolio, which is outside the NIH.
Are you asking for my personal opinion?
Yeah, I'd love your personal opinion.
I don't think we should do research on dogs or cats. Absolutely not. I think it's unconscionable. I understand that there's part of the extramural portfolio that exists that predates you and I.
Yeah.
We are constrained under the law to leave those existing grants in place
for now, but to phase out. That's
but to phase them out, we are working tirelessly behind the scenes. We are doing a very critical assessment of the entire extramural grant portfolio to understand where different types of animals are being used and for what purposes, and to basically create an action plan to phase those out as quickly as possible under the law.
Well, Nicole, I'm grateful that you agreed to kind of spend some time with me. I know how busy you are, given that you run this crazy office, which I still can't pronounce, DP Kipsey, which has so many parts of the portfolio. But I also want to thank you for your leadership over the decades for promoting these alternatives to animal testing, for emphasizing the need for research that really does translate to better human health, because that, of course, is the mission of the NIH. And thank you for your time to talk with me.
It is always such a pleasure to talk with you, and it is such an honor and such a privilege to work for you and for the entire leadership, for the HHS, for the administration. We have so much support in this space. It's truly heartening to see how we are going to transform science with more human relevant methods.
Thank you, Nicole. Nicole, again, thanks for joining us. To our listeners, thank you for tuning in. Drop your comments in the chat as to what subjects we should cover in future podcasts. And please be sure to hit that like and subscribe button on your preferred podcast platforms.